Good afternoon, everyone. Until the last class, we have learned, or until now, we have learned several things about how to construct a recombinant DNA. And in the following classes, we will also learn about several things, including how to analyze or use several molecular techniques, the principles and the ways to interpret them. In that sense, we will learn about um, genetic variation, and later we will learn about how to analyze gene expression. Many of the things, if you're in biotechnology, you might have already learned, but I would certainly want that you have a higher level of understanding. In the sense, you have to come till the interpretation level. So, first thing is, what is a genetic variation? If we have a species and the species has a certain genome, species constitutes several individuals, and they all are variants. All the genes, all the individuals, even though they comprise one species, individuals of one species still are variants. And then on these variants, natural selection can work, and then some of them will survive better than the others. So when you say variants, that means it is about the variations within the genome. So what we are concerned mostly is not, we're not talking about something that is advantageous. We are talking about diseases or genetic predisposition to diseases. And if we want, as you can see from the course, is about recombinant DNA technology in medicine. So if mostly we will be concerned about diseases, how to diagnose them and how to treat them. Those are the two things that we have focused in this syllabus. So when we want to diagnose, we should be able to analyze the genetic variations to start with. That is what we should do. As a part of therapy and treatment in conjunction with stem cells, we will use gene therapy or other alternatives to treat the diseases. So when we want to find out what are the variations, and that is a general thing, you can take even a plant or a bacteria or a human or any kind of genome and try to check what are the variations. If you can recollect from your molecular genetics or molecular biology, you must have learned several types of variations, or we can say them as mutations, right? And there are, at least in two levels, there are point mutations and there are large scale mutations, large scale changes. Did I circulate? I taught molecular genetics in the previous class and previous semester. I think I circulated that in the I Compete GCR. If you are interested, you can watch it. Or of course, there are fantastic videos on YouTube which you can also try on several other authors or teachers. So point mutations, I'll quickly summarize what these are, and then we can think of it. Go ahead. Point mutations and large scale mutations, there are differences in the number of bases that are changed. Okay, if there are 1, 2, or small numbers, then you usually call them as point mutations. But if there are large scale differences between the two genomes that you're comparing, then you call them as large scale insertions changes. Point mutations can be substitutions. To stay GATC, CTAG, say for example, this is a DNA double stranded, and if there was a change, there is a change between this sequence. The sequence in this position it is different, right? The total number of bases is same, but we have substituted, or substitution has happened. In place of A, there is C. Something like C is substituting A, right? So there is a change. And if you remember, you must have heard the words transition and transversion. Sorry, version. Transition is purine to purine or pyrimidine to pyrimidine, right? That means A to G or C to T or vice versa. Transversion is purine to pyrimidine or vice versa. Okay, so that means A could be with C or substituted with C or T, and G could be substituted with C or T. C could be substituted with A or G, and T could be substituted with A or G. Okay, those are substitutions. Insertions, you may have, say, for example, if this is the wild type, we have a sequence GAATC. There is a one base addition here, right? We only had GATC here, but now we have GAATC. That is an insertion, small scale insertion. Deletions would have been, from this sequence, if you are comparing, deletions would be something like this. The A that was present is missing. That is a deletion. When you talk about large scale, of course, I would also want you to think of it as at least this one. You can talk it of single nucleotide polymorphism. Substitutions also can be called as. That means in that one single base position, say if we had a GCT something, this is the wild type sequence. I'm not writing the second strand. In this same sequence, if we have anything like this could be another sequence. Okay. If you see that in this position, single nucleotide base position here, you're seeing 4 different types of bases. That is, you're seeing polymorphism. That is single nucleotide polymorphism, SNP. Okay. Yes. And in the large scale ones, we have, you will have large scale insertions. It could have been gene A is there, and within gene A or somewhere near between that, you may have large scale insertion like this. It could be 100 base pairs, something like that. You can also have deletions, which means the gene has become small because of some truncation. Some part of it has been lost. It could also be inversion. Say, for example, if there is a DNA which has the genes A, B, C, D, and because of inversion, it could become A, C, B, D. That is an inversion. There could also be duplications if we are comparing this way. A, B, B, C, D. These are genes, okay? A, B, B, each of them. Here, B is duplicated. Transposition, as we will discuss more also somewhere, is something, a transposon, for example, coming and between, sorry, A, B, C, D. Here we have a transposon coming in. So the nature of genome is, or the sequence of the genome is constantly changing. Okay, and they can result in diseases. They could be genetic predisposition as well. So before we want to try any regenerative medicine or any kind of those approaches, then we should first understand the nature of the variation. Okay, many a times it's very difficult to find out in which part of the genome the variation is, and what is the nature of the variation. There are 2 problems that we will need to know. In the current days, many things are much more easy because technical developments have improved that we can all of a sudden we can sequence the whole genome and then compare it with some healthy ones and then figure out which are all the problematic positions. So that's a nice and easy way. Of course, it's expensive and technically demanding, but still it is nowadays things are much easier. In the still, these are still some of the techniques that are used are finding out using, we can do localized expression or finding out there are techniques which can find localized variations, and sometimes we can do genome-wide variations as well. Some of the techniques are Southern blotting and RFLP, PCR-based methods, VNTR, direct sequencing, allele-specific oligonucleotide probes. We have discussed this before in genotyping. Genotyping also comes into the analysis of genetic variation, right? Whether someone is homozygous or heterozygous. When we find that, say, in all these variations, we are trying to find out the alleles of a gene, of any gene, the mutated gene. And in one case, we have to also establish if a person is homozygous or heterozygous. Okay, so the genotyping methods, whatever we discussed, are also part of it. And single strand confirmation polymorphism is another way as well. Okay, before we get in, I also want you to remember that there are some techniques can be used to use any of these techniques, at least in some of parts, we need prior information. Without any prior information, we cannot say, for example, we want to design PCR, we want to use PCR-based methods. We should know the sequence a little bit at least so that we can design the primers, right? Southern blotting, if you're saying we should know what we want to look at, and then we have to make a probe. So we need to have some knowledge about the sequence. So as we will also discuss maybe in the next class, where we will try some of the assays, something genome-wide comparisons is where we don't really need to have, any sequence specific, specific sequence of the variation. We can just go ahead and try to find out, and then we'll get a whole huge amount of data from which we will compare and plan the experiments so that we can decide what are all probable problematic genes or variations. Okay? Yes. So basically, what I want you to remember is there is the genome, and genome will give rise to the transcriptome based on the conditions and the signals and all. Transcriptome is in mostly results also in proteome, and the proteome will give the structure or the form and function to the cell, right? So if there is a variation here in the genome, it might reflect in the transcription. It might also reflect in the proteins that are produced. And that is what is reflecting in either loss or alteration in the form and function, which as a part of the body may cause the disease. Okay, so molecular techniques are a very common thing used in, it's becoming increasingly common in diagnosis. Especially in Western countries, it's much more common. Just biochemical is insufficient. If you want to find the molecular basis of a disease, then we have to go for analysis of genetic variation. So for each of the technique, even before also, I would want you to, for the previous techniques too, if we have already covered any, we should know objective. What is the technique used for? Then what is the principle? and what are the limitations and applications. I think if your biotech people may have already learned many things. So I hope we still have some more things to learn, and we should all try to increase our understanding of where we are in this case. Okay, for all techniques, please write these down in yours. I might cover many of them, but some, if I missed it, you can always fill up the gaps. So this is pulse field gel electrophoresis. Okay, I should just give me a minute. Okay, I wanted to, no. Excuse me. Yes. You can see my screen, right?
Yes, sir.
Yes, this is pulse field gel electrophoresis of a GIF, where basically this is where you're loading the sample. And once you have loaded, sorry, my bad. Yes, so we load the sample here, and we have several electrodes. This will be negative, and this is positive, negative, and positive. I think I was describing about pulse field gel electrophoresis when we were discussing about gene library using EMBL3A. So there are different, and at that time too, I said that there are different configurations of how the electrodes are placed. In this system, you have, this is one set, and this is the second set, and this is the third set of electrodes. These are the opposite ones. So the electrophoresis unit is fixed or connected to a computer, which will keep changing the currents to different set of electrodes. We'll call it as A, and this one as A. This we will call as B, and B, a pair of electrodes there, and C and C. So the computer will keep alternating. Why do we need that? First of all, when we want to when we want to electrophorose long DNA, very large ones, then it becomes, I mean, as the greater the size, lesser the concentration should it be, the concentration of agarose. So if the size is too enormous, you cannot go beyond a certain concentration of agarose, because the gel will be too fragile. And because during electrophoresis, heat is produced, because of that heat, we might actually also lose or melt. The agarose might melt easily. So we don't want to compromise beyond an extent on the concentration of agarose. So we have to rely on other things. And one of the alternative is to use these kinds of this pulse field gel electrophoresis. Pulsing means you keep changing a little bit, give a shock again, like that. And here we have A electrodes working first, then we can be, maybe B is activated and C is activated, and so on. Then the pores, even though the DNA is larger, it can still have multiple opportunities to move through because of the reorientation, probably the reorientation of the DNA, instead of getting stuck to one place. So that will increase, and when we electrophorose, we can have gel bands look up as something like this. Okay. So that is about pulse field gel electrophoresis. It is used for electrophoresing large-scale DNA. Oh, sorry. So now the one of the techniques we want to discuss, we want to know is if there are any insertions, deletions, how do we do? This is about large scale ones, and you know about it. It's called as RFLP, or restriction fragment length polymorphism. The meaning of the technique, that is what I said. If you know the everything is in the name, if you know the name, you can figure it out easily. That means that the meaning of the technique RFLP is, if you restrict a DNA, if you restrict two different DNAs, DNA A, B, and C, three different samples, and you are using the same restriction endonuclease. Say, for example, A, 1 second, I will just do something now. So I'll try to explain the word and the term RFLP, and then probably you'll understand it better. It's always important that we associate the name with the principle so that you will be able to understand easily or remember easily. So I have taken, these are all isolated from the same members of a species, sample A, sample B, and sample C. This is the nuclear DNA. And then we are treating all these three with enzymes and with one restriction enzyme, say for example, EcoR1. Okay. If they belong to the same species, they should ideally give the same number of bands of same size. Yes, they should give like this. That is what is expected if they are identical. But as we already were discussing, they are not identical. There will be variants among them. And so if there are some things like this, you are trying to see that restriction fragment, these are the fragments upon restriction, and you see differences, that is different morphologies in the pattern of the restriction fragments. So you call it as restriction fragment length. You are only discussing about the length and polymorphism. So usually this technique, I hope you understood the name, and from that you can always um, derive the principle of it. So RFLP is usually in association with southern blotting. Southern blotting is a simple technique. I will see if I have it here. No. Southern blotting is a technique in which you take I will go to the previous slide here. So we isolate the genomic DNA, then we cut it with restriction endonucleases, restriction digestion, I'll put it as. And then we can do pulse field gel electrophoresis if we require, or normal one also. It depends on the sizes that we are expecting. And then we will do blotting. Blotting means if this is the gel, I'm drawing the cross section. We have a thin membrane like this, and we will have a block that is put on which the gel is present with the papers. And on top of it, we will put nitrocellulose membrane. I'll draw it like this. This is the nitrocellulose membrane. And we have, this is all in a trough with buffer. Okay. The buffer fills it up. And we'll put a lot of paper towels on top of this, on top of it, to create some kind of capillary action. So the buffer from the trough will be taken up. It will through wick, as a wick it will go, and it will start moving upward direction through capillary force. The DNA after electrophoresis is present in here. And that DNA will now, because the buffer is moving along with the buffer, the DNA will move upwards. And what is it encountering? It is encountering a nylon membrane or nitrocellulose membrane. And on the nitrocellulose membrane has high affinity for DNA, because of which DNA is now blotted onto the nitrocellulose membrane. If we spill something on the floor in chemistry lab, for example, you are supposed to take a blotting paper, unless it's not very dangerous. you can use the blotting paper to just absorb the water and then throw away the blotting paper, right? Spills can be easily maintained that way. Similarly, we have blotting here. The DNA is getting picked up by the nitrocellulose membrane by the arrangements that we have made here. Okay, and then we need to, so once I'll try to draw the nitrocellulose membrane here, and the DNA is fixed on top of it like this. Okay, now we are going to add we have to figure out which sequence is going to, which part of it is, whether the sequence is present or not. First thing is about that. If we are doing southern blotting, the objective is to find if there is a sequence of our interest that is present in the sample or not. And how would we know which is which is the sequence and how to find it out? We have to use a probe. The probe should be radio labeled, or there are other methods, but usually radio labeling is the common one that everybody uses, majority. A probe is a DNA, and you can sometimes RNA is also used, which is usually single-stranded. Okay, and it is complementary to the sequence of our interest, right, or gene of interest. So we are going to now add this probe to the nitrocellulose membrane, and then the process of hybridization will be allowed to happen. Hybridization means base pairing, basically. Okay, there are other processes wherein you have the DNA double-stranded, you would have baked it at 80 degrees or even higher, and the buffer conditions are maintained such a way that the double-stranded DNA would have become single-stranded and gets fixed to the nitrocellulose membrane. So the nitrocellulose membrane, as I said, has high affinity for DNA. And now we are going to, or nucleic acids also in general. If we are going to add a probe, which is also made up of nucleic acids, the probe can bind anywhere on the nitrocellulose membrane. So prior to addition of probe, we usually block the whole membrane, so that if there is anywhere it is base pairing, it shouldn't be because of the phosphate backbone, but it should be because of the hydrogen bonding. So the probe should have come and base paired with the DNA along with its radioactivity. Okay, so that is how hybridization would occur. Hybridization between the probe and the target sequence. Okay, and once you do autoradiography, you will find out one band coming up based on how many copies are there and so on. In one lane, one band. If there is, say for example, there are three lanes I drew here. I'll call them as A, B, and C. Here is A, B, and C. And this is what happens. That means A does not have the sequence that we are looking for. Sample A is not having it. Sample B is having it, and sample C is having 2 copies of it. That is the interpretation that you can make from it. Okay. We can also have, when we are interpreting, we will get different possibilities are there. And that is what we are looking at in the next slide. So they have taken 4 different samples, say 1, 2, 3, and 4 samples, and each of them they are already telling what is the difference. And here is the probe. A single, this is a point mutation, loss of a restriction site. There is an insertion in one of it, and there is a deletion in another one. Okay. In each of these cases, we will consider the first one as wild type or the normal one. and the probe is complementary to the region that we are indicating here. So I will also circulate one assignment that, or it's kind of exercises that everybody should do, I think. And once you have finished it, you'll feel much more comfortable about it. If you look at a gel like this, first one we are continuing, concluding that as wild type. First one, sample 1 is wild type. There are sample 2, 3, and 4. What does this indicate generally? Normally, if you are doing electrophoresis, what would you say? In electrophoresis, the mobility of the nucleic acid is dependent on conformation, and also if it is linear, then it is size. Larger size will travel slower than that of the smaller ones. Smaller ones will travel faster, right? But when we are doing restriction length, fragment length polymorphism, we have taken DNA, we have digested it with restriction sites, restriction enzymes. So in it, in RFLP, if we get this is assume this is the wild type, and if we get a band here, what would that mean? Does it mean that that gene is large or huge? Not necessarily. That is what we are trying to understand here. So
It could be a
let's
possibility that they are not in relation with each other, or it's different from one another.
which one um, which one is not in? Say,
I think
first
one
of
you
all, these two are sequences, right? If the probe is binding, this is after southern blotting, and probe is binding, then the sequences are same. Okay. Only thing is associated with a larger fragment or not is a question. Okay. Ajit, is that what you're saying or something else? Did I miss it?
I was meaning that the sequences I think are similar, or we are trying to study what are the slight variations in that. I think so. Like, are they related to one another or something like that? I was asking, not the size aspect. I was not asking.
Okay, okay. Yeah, I'm trying to, at this point, try to discuss the size part, okay? Because we can always be confused about it. Say, for example, here is the probe. I will say this is 2 kb and this is 4 kb fragment, okay? And here is a restriction site, here is another restriction site, and here is another one. In each of the cases, it is, say, eco R1, okay? So eco R1 is in first sequence, we will put it, so here is GAA, TTC, GAA, TTC, and here is another one, GAA, TTC. So I'm not writing the second or the complementary strand, assume that it is there like that. So if we add, also take this as trying to put it in sample 1, 2 kb, and this is as 4 kb, distances between the restriction site. I think we also have a lot of restriction mapping to do. I have to post them as well. So if we add restriction endonucleases, eco R1, then we will get 2 fragments. This is 2 kb fragment, and somewhere here we'll get 4 kb fragment. I'm not talking about southern blotting at this time, okay? Just cutting wise, we will see. In a different variation to this, we have another DNA. I think it's better to draw it in a different color so that it will be easy to identify. So if we had GAA, TTC, and then GA, TTTC, then GAA, TTC, and this complementary strand. There is a difference between the wild type and this one. Now, if I cut the sample 2, and this is sample 1, I got 2 and 4 kb fragments. If I cut sample 2 with eco R1, I would get it. Echo R1 will cut here. It will not cut here, but it will also cut here. So the fragment size would actually be 6 kb, right? Counting from here, 2 plus 4 kb. It's you can also see the other part of it. I will try to describe what else might also result a larger fragment. Wait, we'll come to that in a few minutes. Just look, let's finish this one. So I hope you understood that. This is a single nucleotide polymorphism in one base position. Where A should have been there is T, because of which it's no longer EcoR1 site, and therefore EcoR1 cannot cleave there. And so a large fragment, restriction fragment, is there with the same sequence except in this one base. That is a single nucleotide polymorphism in that base. So it is likely to give a larger band like this.
So,
Yes.
if you understand it in simple words, rather than saying in some terms of size, since you've mentioned that the band where it's a little higher doesn't necessarily mean it's of a similar, I mean, of a higher size. So I would rather say it as a maybe not cleaved at that site where it should belong, rather than the same enzyme didn't cleave at that particular site. It should have cleaved because of the SNPs.
Yes, I agree. That's fine. Yeah. But we can also, we will say take sample 3. In sample 3, we have everything same. GAA, TTC, GAA, TTC. But there is something difference. We have and another GAA, TTC. But there is also a large, say for example, there is an insertion here. So the original size here is 2 kb. Assume that the insertion, this is 2 kb from here and 2 kb from here. Okay, that is 4. And now we will take it as another 4 kb that has got integrated. Just take it for that, for the sake of discussion. Now if we add EcoR1, okay, and then what size of bands would we get? We will get a 2 kb fragment. We will, because from here and here, and then we will get 2 plus 4 plus 2, that is an 8 kb fragment. This is 2 kb fragment. So the band would then be, if we are comparing here, let's compare again what would happen to sample 1, sample 2, and sample 3. Sample 1 will give this 2 kb fragment and this 4 kb. Just electrophoresis. Don't talk about southern blotting at this time. 2 kb and 4 kb. Let's take sample 2. There is a cut here and there is a cut here, but no cut in the middle. So we'll get a 2 plus 4, we'll get a 6 kb fragment. In sample 3, there is a cut here, there is a cut here, and there will be a cut here. So we'll get a 2 kb fragment. And then because there is an insertion of 4 kb, the total size of this would become 8 kb. So we'll get a 8 kb fragment. Okay. What I'm trying to elaborate here is that when you see a band that is up, this is the wild type, right? If that is the wild type, if we see a larger fragment than the wild type, does it mean it is an insertion, or does it mean it is a loss of restriction site? There are two possibilities, and I want you to look at the experiment properly every time a question is given, and then explain, interpret it correctly. Okay. So with with regard to the probe, we will do an exercise. Okay. And then let's see if we get to it. And all the techniques we are going to go in greater detail. So I would want that you pay attention and try to understand it and acquire the skills to is we have 15 minutes, so we will just use it for as a quiz. We can all try to do it. I'm going to draw a sequence, and I'm also going to say, okay, I have, this is an EcoR1 site, EcoR1 site, and EcoR1 site. And the probe that I'm choosing will cover from here to here. Okay. That means the probe can bind to this part of the sequence as well as this part, the second part. The first one we'll say is as 2 kb, and the later one is 4 kb. Okay. This is the same setup as what I am choosing here. I will try to erase it and we will see. In this case, we have a probe only in this space. Even though it is cut here and here and here, only this place will show up on the northern blot. Southern blot. Why is that? Because probe is specific, will hybridize only to the 4 kb fragment and not to the 2 kb fragment. And that is why we can only see one band. Okay, I hope you understood it. What I have described before is theoretical, and in electrophoresis, if you just take DNA, genomic DNA, and electrophorese it, you will not see any specific bands. Mostly you will see some smear-like things because there are random cuts. Only upon addition of the probe, you might probably find one band as in the autoradiogram. So in the second one, there is a loss of restriction site. That's why we see a larger fragment, about 6 kb. And the third one, there is an insertion. It's probably not so much, say this is 4 kb and this could be 1 kb. So around 5 kb fragment. And in the last one, say 1 kb is lost or so, or maybe a larger fragment is lost, say 3 kb is 3 kb deletion. So what you're left with is 1 kb. Okay. What I was trying to do here, we will do it here itself, and tell me what would happen when we do something like this. So the whole experimental setup is almost same. Everything is same. I'm only changing one part. The part that I'm changing is the length of the probe and what it is going to bind. I've changed the length of the probe, and that is the same for all. Okay. I hope you understood what I meant here.
Yes, sir.
Yes. So let's see. What would you expect in the bands? So here is sample 1, 2, 3, and 4. What bands will you see in for sample 1, 2, and 3, and 4? We will assume this as 2 kb and this as 4 kb. Insertion, I said, is of 1 kb insertion. Deletion, I said 3 kb deletion. Yes.
We'll see 2 bands for sample 1.
Why?
Because even after restriction, there'll be 2 different fragments, the 2 kb fragment and the 4 kb fragment, and the probe can bind with both of them. So we'll be able to
That's
visualize
right.
both.
That's right. So if we did not have a probe as I drew, and only this much part, then it will only recognize only the 4 kb fragment, and we will not be able to see the 2 kb fragment, right? So if we, like we had before, if we have it like this, then the probe can bind to 2 kb fragment as well as 4 kb fragment. Okay, in sample 2, what shall we expect?
I think it's the same what we've seen in sample 1.
Why? Why is that? What is the difference between sample 1 and sample 2?
Shouldn't
Loss
that
of
be
restriction site.
So why would it be same as sample 1?
Sir, in that case, then we'll see a 2 kb fragment 1 and then a 6 kb fragment, since there's a loss of restriction site. Like I might have said that a mistake. I made a mistake in that. I think,
Yes.
like if I'm not wrong, a 2 kb fragment and a 6 kb fragment.
Why will
Since
you
there's
get 2 kb fragment?
So since there's a probe already, sir, like we have a probe in there.
Yeah, you didn't get a 2 kb fragment. Yogeshwaran, you're right. Only a 6 kb fragment, isn't it? We have, this one is not getting cut. The size from distance from here to here,
Yes, sir. Yes, sir. It's only
it
6,
is
sir.
only 6 kb.
Yes, sir. Based on the first one, I've just continued.
Yes,
with.
yes,
That's
yes.
a
That's
mistake,
all
sir. Yes,
right.
sir. Only 6 kb, sir. Only 6 kb. Sir. Yes,
That's
sir.
all right. Yeah, that's okay. So what is with 3? What is with sample 3? Anybody?
one 2 kb fragment and another 5 kb fragment?
Yes, 2 kb and 5 kb. Why is it 5 kb? This is 4 kb fragment
Because
and
of the
an
insertion
addition of,
process.
that's right, addition of 1 kb. That is 5 kb fragment. Okay. Please follow this because I'm going to come up with a complication for the next one. And 4, what will happen in 4?
We'll have a 1 and
That's right. We
1
have,
and 2.
sorry, this should be 2 kb, right? Yes. This is,
So that one is 2 kb. This one
that's
is
right,
1.
1 kb, and we have a 2 kb fragment. Yes? Is this clear?
Yeah.
Okay. Now
Sir.
I'm not going to tell the detail. I'm not going to tell the detailed picture. You can draw it on your paper and try to come up with the answer. Okay. So I will erase this. Yes. Every setup is like this, and the genome that I'm giving here is wild type 1. Allele 1 is wild type, allele 2 is, and 3 and 4 are mutants. I'm going to complicate it now, so try to come up with correct answers. Let's see if we can make it. And I would appreciate, I already appreciate that you people are participating. It's a nice thing, and I want you to do it a little more. I'm giving you 3 different samples. Sample A has allele, is homozygous for allele 1, B, C, D. Sample B is heterozygous for 1 and 2. Sample C is 2 and 3. Sample D, sorry, is 3 and 4. Can you tell me the banding pattern? Sample A is wild type or homozygous. Okay, we know that we are all diploid, right? Here we are only showing about one sample, one allele probably. Diploid means we should have 2 copies. What if, like the genotyping experiments that we have done? So here I'm saying setup is same as this, what is given here. And I'm saying the sample A is homozygous for allele 1. What should we expect in the banding pattern after southern blotting in RFLP? Yes.
Sir, if I'm not wrong, for the first one, we'll just have a 2 kb and a 4 kb fragments just alone, since it's homozygous already.
Okay, that's fine. Yes. Sample B.
Sir, for sample B, we'll have a 2 kb, a 4 kb, then a 6 kb.
Everybody agrees? Or do you have any contradictions to that?
2, 4, 6, sir. Yes, sir.
Okay. 2, then 4, and from the other allele, we'll get 6, allele 2, right? So what about C?
2, 5, and 6.
2, 5, and
Uh,
6.
okay. 2, this is 4, say, we'll say 5, and 6. Is that correct? 6 is coming from allele 2, and from allele 3. Okay, I will not write that. It'll get confusing, so I'll just leave it there. But I hope you remember. I'll write the fragment sizes instead. This is 2 kb, 4. Here will be 5, and this will be 6. And what about D?
The
1,
1
2,
kb
and
fragment, 1, 2, and
One,
I think
1. Uh, yeah. Tell me.
we'll get a 2 and then a 5.
2 and 5. Yes, that's correct, I think. Okay. So this is how we play puzzles in the exam. This course should be filled with puzzles. Either gel picture will be given and ask you to determine what is the nature of the genetic variation, or I'll give you the genetic variation and ask you to draw the gel picture. You will never get a question in rDNA in medicine as what is southern blotting. That's not going to happen. We are supposed to reach a little bit higher level and try to be in a situation where we can interpret the results. Okay. Do you have any questions? I thought I will cover more, but I didn't. So we'll see if we can go faster in the next ones. But it depends on how much you're contributing, your understanding. You should probably be able to ask questions if there is any difficulty, and then we can all sail together faster. Okay.
Yes, sir.
Yes. Any questions from whatever we have discussed today?
so this is just a request.
Yes.
So shall we have the gel picture that we drew at the last as the question, and shall we try to figure out if sample A is homozygous or heterozygous? Like, shall we do it
Yeah,
in the
but
reverse?
now you will know it, right?
Like a different question, sir.
Oh, sir, similar to it,
Yeah.
sir.
If it were an
Not the
exam,
exact one.
that's what I would do actually. You will get the gel banding patterns, and please, everybody should realize that all questions will be like this. So you following is very important. You want to do it now or later? We'll do
Maybe
it
if you could
later.
give us a question. Yes,
Yes,
sir.
say it again, please.
So if you could give us a gel pattern, we would work out and maybe update in the group.
Yeah, that will be nice. Okay, I will. No, I actually want you to also do an assignment that I do every year. Okay, especially if you are taking genetic engineering course and the lab. I used to be involved in lab as well. In that lab where I take, it is a compulsory that everybody submit that assignment, and it's a very good exercise. It's a from a lesson, I think. I'll try to circulate that. I would want that you all try to solve them, discuss about it, and learn those things as well. Yes, I'll try to get some practice questions like these, and then you can try. Okay.
Thank you, sir.
Yes, good.
Okay, sir.
Any questions or so? I thought somebody will ask what would happen this loss of restriction site if it is just one base star activity and something. It is also a possibility, but we will usually people will, when they are optimizing, they will try to optimize a certain genetic variation, or I mean, they will optimize the correlation of a genetic variation with that of a disease, and the sequences, the restriction enzyme that should be used, and the fragment length, the probe, and so many other things they would have optimized. Or even if you are doing it, you would have tried with different probes. Previously there was one probe, and then I have modified the probe. If there is probe on one side, you will have a different banding pattern. If you changed it to a different one, you change the location of the probe. Say the simplest one would be to do this instead. I will change the whole. Everything is same except that the probe is now moved on to this place. Okay. Then what kind of banding pattern would you get? And how will the banding pattern be different from, say, this is probe X and this is probe Y? If only probe Y is used, what kind of banding pattern would you get? If probe X is only probe X is used, what kind of banding pattern will you get? Okay. Will you be able to identify the genetic variations here? This can be identified, but these might not show up at all, isn't it? So that is how then they will think that, okay, probe X is not so great for our analysis, and so we will use probe Y. That is optimization part of it. Yes? Any questions?
So I have a doubt.
Yes.
So how do we know if there is a genetic variation at all in the first place to make sure that we design appropriate probes? Like there might be more variations,
There
but
will
we
be a
might
lot
have.
of variations. Yes. That is why, I mean, I think I understand the question that you're saying. That is why I said here, we should have known prior, we should have had some prior information. Those usually come from techniques like genome-wide variation comparisons. You can also have karyotyping. You heard about it, right? Trying to check the banding patterns on chromosomes using, I think I'll try to see. You
If
can
I'm not
also
wrong, isn't it microarray technology?
say it again.
If I'm not
Please.
wrong, isn't it microarray technology?
No, no, no. Karyotyping is about trying to see the chromosomal aberrations, aneuploidy, polyploidy, and also if there are any large-scale inversions within the chromosome. So typically, if it is a genetic disease, they would have taken the DNA cells of the person. They would grow the cells, and then they will splatter the cells using many techniques, and then the chromosomes will be spread out like this here on the glass slide, and they will watch under the microscope. These are olden techniques, okay, old techniques. And then we will be able to figure out if there are any chromosome large scale banding patterns. We'll compare it with the wild type, and we will know if there are any large scale chromosome losses, inversions, deletions, or additions. And then we know on which chromosome and which part of it is affected. Previously, you should remember that we know nothing about the sequence. We only have chromosomes. People were only working with banding patterns. And as the advances came, we now know a lot about sequences, and we know we are in a situation where we can directly go for genome sequencing. Okay. So this is whatever we know right now is built on the information that we got from a long centuries, or sorry, decades of trying to find decades of research that um, went through a lot of logical progression. You should remember, I think I will just stop with this 1 minute. Okay. There are models and there is technology. So there are two types of research. Both are, of course, intermingled. Okay. Some research is purely or mostly based on the technology. Some research is based on modeling and theoretical thinking. Okay. Nowadays, if we know, majority of the science or research is taken up by the technology, NGS, and you can take many of them. You have large scale technology or data handling and other things. You can just do those and you'll get some information. In times of uncertainty where you don't know what to do, that is when theoretical modeling will come about. You think about stuff, you try to reason logic, and try to build some models from which you will try to get to the answers. So my job is, while I'm trying to discuss about the technology, my interest would be to inculcate a theoretical thinking and modeling into you. And that is the reason why I'm spending time on analysis, interpretation, and other things. So I would want that you follow it as well. And if you have any questions, please come up and ask me. If you're shy, you can always message as well. And you can think, oh, did you know about that CRISPR technology? Oh, did you know about NCS technology? They're all technologies. And there will be a lot of progress that technology can take away. But it doesn't necessarily have to improve you as a thinking person. I think, in my opinion, okay, we can always differ, that the theoretical and models-based research and thinking is what you should improve, because anybody can do this one. It is just, every technology is kind of, you should know the principle and then use it to run it, right? So I want you to think about models. When nobody knows anything about anything, how did we get to where we are? And that comes mostly from the people thinking about it. And that is one of my ideas in what I want to inculcate through this course. Okay? Sorry, it took a little longer. Any questions? Or so you can always WhatsApp and message, and then we will discuss further. Okay?
Yes, sir.
Thank you, everyone.
Thank you, sir.
Thank you, sir.
Yes, see you. Bye-bye.